Okay, so it's 9 o'clock. So I think we can pretty much get started. Hi everyone. Welcome to today's Immune Zoom seminar. So I just want to, before the seminar, I just want to emphasize one more time. It's the summer season now. We actually have 2 seminars per week, and one is within the week. Usually is Thursday in Beijing time, and then once weekend, usually Sunday. So today it's really my great pleasure to welcome today's speaker, Dr. Wenfei Jin from Southern University of Science and Technology, and from Shenzhen in China. So a very, very brief introduction about Wenfei. Wenfei currently is an associate professor in the Department of Biology in Southern University of Science and Technology in Shenzhen. He actually received his bachelor degree from Zhengzhou University and then continued to pursue his PhD in computational biology from Chinese Academy of Science and Max Planck Society Partner Institution for Computational Biology. And then from 2012, Wenfei actually started to pursue his postdoc training, actually in NIH, precisely with Dr. Kojitsa's lab, who actually just gave a talk last week. So after a very productive period of postdoc training, Wenfei started his independent research program as assistant professor in South University of Science and Technology from 2017. So Wenfei's lab actually particularly interested in a couple of different aspects of genetics and the computational biology. In particular, he's interested in single cell sequencing, which I think we're going to hear a lot about today, and then how the single cell sequencing can be applied in cancer biology. And then second thing is bioinformatics, and then identifying genetic and epigenetic changes that drive oncogenesis. And lastly, his lab also interested in developing cancer prediction and treatment module for precision medicine. So Wenfei has been focused on developing genetic technologies, particularly developing single cell technology, which actually have a lot of great tools from his lab, including single cell poly-A sequencing, ISS AAC sequencing, CCI, single cell DNA sequencing, and then KSSD. Actually, he has been very, very productive and then published a lot of including methodology and then biology research, single cell genetic research result on different journals. So based on his work, he's been receiving of many honors and awards, which including the National Young Talent Award and Puro River Young Talent Award, Southern University of Science and Technology Excellent Postgraduate Supervisor Award, Springer's CSS Award, and CAS Excellent Doctor Dissertation and the Dissertation Award. So I think without further ado, Wenfei, welcome to our Immune Zoom seminar, and we are very much looking forward to your talk. Please go ahead.
Okay, thanks, Chuan-Wu, for the introduction. So yeah, first I give this topic because my background is genomics. So I first talk genomics, then I talk some its application in the tumor especially tumor immunology. Okay. So yeah, before we talk single cell sequencing, we first talk about the cell heterogeneity and the single cell omics. So firstly, we know that the cell is the basic unit of life. So when we do study, we use a lot of cells, but that's where it's provided it's already a lot of biology inside. But today we talk at the single cell resolution. So when the first single cell technology show up, that is because especially at earlier time, usually like a sperm or something that's especially in the earlier developmental, usually you only have one cell, then that's you need a single cell technology. But later, sometimes you still have a lot of cells, but whether we still need a single cell technology, that is at the earlier times we usually using population average assay. Now it's still dominant approach. So usually you have a lot of cell, you make one average value for the total cell. So but once there's heterogeneity, then the results may be not so reliable, especially sometimes when different population have different value, then the results may be misleading, and especially we lost the information over each cell. Then the single cell sequence developed very quickly, especially single cell RNA-seq. There are a lot of technology like including multiplexing, microfluidic, and and droplets, pickle, different technology, and the throughput of single cell sequencing improved quickly. And also not only single cell RNA-seq, there's a lot different single cell sequencing technology. Like you can study the hidden modification, mRNA. It's also you can study the regulation and even 3D genomics. So single cell sequence a lot different technology depends on what you want to study. And then so we first introduced one technology that when I was conducted in Keji's lab, that is single cell DNA-seq. That's especially that the DNA-seq is a very powerful technology for detect chromatin accessibility many years ago, and usually it indicates the regulatory elements. So at about 10 or even long years ago, we at that time we wanted to detect chromatin accessibility at single cell level, and in this study we add a circular DNA and then add a buffer, then to significantly increase the number of reads in each cell. So then we can really capture the chromatin accessibility differences between different cells. And in this study, we found the housekeeping gene usually with much higher accessibility, and tissue-specific gene usually with lower accessibility and higher cell variation. And for the promoter usually with much lower cell variation, but the enhancer we have distal regulator usually with higher cell variation. And at that time, we also studied the FLP by the modified single cell DNA sequence technology. Then we detect the tumor-specific THS, the tumor-specific accessibility. And we also identify the specific motif. But one of the motif is potentially influence the binding of P53 on the. So then we expected that because this mutation will reduce the binding of the P53 and then reduce the expression of TXAO1. It's a regulated gene, target gene. Okay, then later our gel shift assay supported that this mutation really reduced the expression of this target, and also the promoter assay also indicates the mutation lead to reduce the activity. And also our collaborator, the other member in the lab using similar technology, but using change the enzyme to amylase detect the nucleus amplification. So now I'm introducing one study in my current lab. So before we talk this technology, we call single cell poly-A-seq, we talk about the smart-seq. That is very commonly used in this field. It's can start the 4-long single cell RNA-seq. It's with temporary oligo switch oligo, so can capture 4 lengths, but that is 4 lengths usually cost is high, and could not really provide the poly A information. So we do some modification to increase especially enrich the reads at the 3 poly A sites, and especially amplify the 3' region. And then we indicate it's because this is 3' you can similar as 3' RNA-seq. Then we compare the 10x, we found it's indeed capture much more reads than in the 10x single cell RNA-seq. And then we also check whether they really is good for study the poly A site at the single cell level. Yeah, because we especially amplify the 3' and then we can get a lot of poly A supporting reads. Then we can directly detect the poly A site, and we based on we can see that this time indeed detect a lot of poly A supporting reads, and it's especially enriched the read special near the end. That means near the poly A site compared to the other technology. Then we also can use this direct the rates directly capture the cell type using the. Then we analyze the poly A site usage, because a lot of genes with more than 2 poly A site. Now our analysis shows that even we found the gene have multiple poly A sites, but at a single cell level, usually only one cell prefer use only 1 poly A site. That means although at the bulk data level, you see the poly A the gene using the multiple, but in single cell level, most of the time they only use 1. And especially we found that cell circle is especially enriched the poly A site variation gene. That means potentially cell circle is highly associated with the poly A site usage. And then we compared the poly A site usage between the cell sequence, and we found that indeed the poly A site changed a lot among those cell in different cell states compared with the gene expression changes. So that's poly A site change is more dramatic. And also we found that at different stage the poly A site usage change is much more than the expression change. So that means poly A are more likely to change during the cell cycle. And then we put all the gene that with poly A site change together and do a cluster. We found that 6 group of genes, they show different, each have their specific dynamic process. So during the cell cycle, and especially each of these cell category, the gene category, we enrich different gene category. That means each group of gene show different feature that may play different role during the cell cycle. So that supported that poly A usage change is important in the cell cycle regulation. Indeed, we like, for example, we see the gene like MSL1, LCPDH, their poly A site usage is strongly associated with the cell cycle. That means cell cycle really is important for role in the associated with the poly A site usage. And then we found that once we knock out one of the poly A sites, the cell cycle will slower usually, and sometimes it's faster. So that's supported that poly A site usage is pretty important in cell cycle. Also, we also analyze the poly A site usage with the gene expression regulation. We found that it's indeed most likely they are more like negative related with the gene expression regulation, gene expression. Then we talk another technology we call the SAXIC. That's is a single cell multimodal omics. So because either RNA-seq, poly-A-seq, or some most of the sequencing you only carry one modality, but nowadays people want more information, so then we usually want to capture more modality. Here we want to capture both chromatin accessibility and gene expression, because we know that we already have some experience on chromatin accessibility and gene expression. So indeed, there's before our study, there already technology that can capture both chromatin accessibility and gene expression, but usually those protocol are very it's very the protocol is long. You need to do a long time prepare library. But at that time, we are lucky found one paper about cover later on PNS, and this is a technical called Sherry, because in traditionally when we doing RNA-seq, we usually doing RNA-seq, we need to capture all the RNA, do RT, then get cDNA, and prepare the library. But in this technology called Sherry, because it's using the new finding that the TIN5 can capture can target the DNA RNA hybrid. That means we do not just go. We can reduce some of the steps, like when cell license, then do a RT, then now you have the DNA RNA hybrid, then direct targeted it for make the library. Then in this case, you reduce a lot of steps for prepare the RNA-seq library. And then we know that a TAC-seq is usually using TIN5 tag the DNA. Now we combine two to make 2 times TIN5 segmentation. First, we get the nuclear isolation. Then we using the TIN5 to tag the chromatin accessibility. Then do a reverse transcription. Then get the DNA. This time we get DNA and RNA hybrid. Then do another time tag documentation in different index. Then now we get both. Once by the 2 step, then we sorted the cell in either by fax or using droplet or some other technology. Now then prepare a library. Then we can capture both RNA, single cell RNA, and single cell attack-seq information. Then so this technology is we call the 2-time segmentation. The first time you get the chromatin accessibility, the second time you get the RNA. Then it's not the protocol is very simple, and the ideology are simple. Then we analyze some cell to indicate the technology. So once like on the cortex, then we found that either the gene expression can class the cell, and also chromatin accessibility also can class the cell. So they are most of them are consistent with each other, although the shape may be a little different. But when we check carefully, we found that one of the cluster is only one cluster in the RNA-seq. It's EXL4 file, but for the attack-seq, it shows 3 clusters. So whether the 3 clusters are different, then we do some analysis, found it's have some difference, especially each cluster how they have specific accessibility peaks. That's because we already found there are 3 subcluster, and each cluster they have specific enrichment motif. So that means the potentially this are biology meaningful. Then we analyze the pseudo time, then found that they indeed some difference. Intriguingly, that we found that for this cell, the RNA-seq, like here, for example, is more likely more similarity higher, but for the attack-seq, is changing more dramatically. That means indicated the chromatin accessibility is more heterogeneous than the RNA-seq. And then we developed another approach to analyze data, because previously we developed an approach to generate the data, and now we analyze data. One we call the single cell graphic autoencoder because this is for dimensional reduction. Dimension reduction is very important in single cell analysis, like data visualization, cluster trajectory. But although there are a lot of approaches, like especially t-SNE, UMAP, and a lot of approaches, but we developed this approach by using we call the graphic autoencoder. Autoencoder is rather well used in single cell dimension reduction, but graphic, that means you can know the relative relationship between the different cells. Then that is here we have a graph. Then this will provide more information, and then based on this, we think because the graphic information integrated in this, we can provide more information among the cell-cell relationship. That means can be potentially preserve the long distance relationship among the different cell groups. And this is simple because it's model free. You just input the data because it's deep learning based. Then you can get the results. Then for later, it's okay. Very friendly for visualization, cluster, and trajectory inference. So, for example, when we conduct the simulated data, we found our LCG perform well. The area is for clustering and linear inference clustering. And for example, especially when data that carries cells for the t-SNE, you find some exactly same cell type, but for different states, they are separated very far from each other. But using our SCGAE, that we can make them look very close. They look very close. Like here is the activity and the quotient. They are very close, but for the t-SNE, they are far from each other. Okay, we also developed what we call the compact component index to get to matching the leukemia or cancer on their lineage. For this study, firstly, we usually get make a reference. That means make the linear inference. Then, if we have cancer cell or any other cell, then we're using the base approach, mapping those cells onto this lineage. Then we can infer whether this state or the leukemia or cancer cell. For example, if we mapping the cancer cell on the root of this lineage, then means that this mutation or develop much earlier. Then the cancer cell may be derived from the earlier stage. Like if it's located on one lineage, that means this mutation happened on this lineage. That means we can infer the when and what stage the cancer happened. And now we are moving our immunity and tumor study because it's most of our audience may be working on the immune. So first we talk hypothesis, the hypothesis. It's we know each day we generate a lot of blood cells by hypothesis, and there's a lot of different hypothesis model, like either stepwise process that each family is different into different states by different steps. But also there's like a cloudy model. That means all the cells gradually differentiate into different lineage. So when we conduct a single cell analysis on the CD4 cell, this is usually indicated is hepatotic stem and progenitor cell. So we found they are indeed linear. It's that means they are they do not have distinct subset. They usually are continually distributed. And then we infer the lineage. Then we know that the different lineage sort the cell and into different lineage, that gradually change. And then that is based on bone marrow. Then whether if we integrate more tissue, like we know for blood, we always also have a lot of hepatotic stem and progenitor cell. So we also using CD34 cell to the analyze. Then we found that indeed this is similar as our hepatocyte lineage in the bone marrow, but now we can say this is bone marrow that is similar as the one we showed before. And then peripheral blood with mobilization and peripheral blood and also thymus, that's especially for developed T cell. And we know the cell population are different, especially for thymus, but for the other is usually, although the population may be changed, but still there are a lot of consistency. And then we found one of the dramatic difference between the bone marrow and other the peripheral blood is the B cell progenitor, because for the CD34 cell, the bone marrow contain a lot of B cell, but for the peripheral blood and they do not have B cell. And also we found that for the short time, you can see the bone marrow, they are at a much earlier stage, and the peripheral blood, although they both are CD34 cells, but they most of cells in later stage. And okay, so we were interested in because only bone marrow with the B progenitor, but the peripheral blood do not have. Then we want to do a comparison. We found that indeed the bone marrow will lowest the migration score, but the mobilize the peripheral blood usually with the highest the migration score. And then we compare to analyze this cell how interacted with the stromal endothelial cell. We found that the stromal cell really have a lot of signal with the hematopoietic subsets. That means the stromal cell may be pretty positive in the cell migration. Then we detect that their interaction, especially the interaction we call CXL4, CXL12. This axis, it's strongly interaction with the bone marrow stromal cell with the pre-B, the progenitor or B cell. So that's a strong signal that indicates maybe this interaction will make the B cell stay in the bone. And indeed, if we see the signal, we found that we analyze the signal. We found that the stromal cell in bone marrow is strongest signal, signaling of the signal. That means CXL12. The receptor that is strongest is the pre-B. The pre-B is the strongest signal receptor. That means they should hide by CXL4. Then we do a cytokine assay that found indeed the bone marrow cell will imicate with strongest migration when there are CXL12, and the cell with the higher level of CXL4 will will move faster. And then this model is that the hepatocyte that is mainly developed in bone marrow, but they can more likely to move to the peripheral blood, even and most any stage except B lineage. And this peripheral blood, especially from the T lineage, they move to the thymus. Then we analyze the T cell subset. Then we know for fact sorting the here we max sorted the T cells, either it's CD4, CD8, and then you do a single cell cluster. Your single cell cluster is independent of this fact sorting. Then that means we can see their relationship. We found that the naive cell sorted by that sorted is very usually very pure. They are indeed clustered into the same cluster in a single cell level, single cell RNA level, and also naive CD8. So, but the other cell type is usually Yeah, because they are more heterogeneity except the naive cell. Okay, so there's one subset we call the SAG, that the interferon signaling associated gene. This is a small subset, T cell subset. It's only 100 cell, but they have very strong signal expression the interferon related gene. And indeed, the GO also shows that this gene strongly associated interferon. Either it's type 1 interferon signaling, defense response to virus, or response to virus. Yeah, it's interferon signaling pathway. And then we found that the BST2 high expression in the SAG high T cell. Then we use this marker to enrich it. Although it's still could not really get pure BST2 because many different cell also expressing it. But at least the total the SAG cell percentage is significantly increased after we started using the BST2. And now we do the analysis. If we do not consider this SAG signaling, then we only use all the genes except this cell, this SAG signaling. Then we found that the SAG high T, they are everywhere. They because you do not use this information to cluster the cell. Then if we only use the SAG signal to do the cluster, we found that this cell can be made cluster. So that means this gene cluster, this cell cluster, this cell subset is mainly because they are highly expressed this pathway. So this pathway became a cluster. Then we do the further analysis to the T cell activation using CD28 stimulation, CD3 and CD28 stimulation. Then we found that the BST2 high, they respond much faster than the BST2 minus. Then another case is that if we do the stimulation, we found that resting cells, they are okay, they are in one side, but after stimulation, many cells, their state shapes, they have more broad distribution, they have more states. Then we do the annotation, found originally the resting cell like T naive, T memory, but then do the activation, they more likely to the feature as an effective T, so those upside more likely to be effective. Then the linear indicates they are either from the naive to the effective T or from the memory to the effective T. Then we found another cluster. We called it HSP high T. This cluster is also a small cluster. It's highly expressed HSP region like HSPA1A, HSA1B. That's a lot. So most of all the genes are HSP related, also including DNA, GB1. So that means this cluster we found that most of the HSP high show up after stimulation. In resting cell, there's almost no this cluster. So this cluster indeed, almost all the genes in this gene is HSPC gene. And indeed, the enriched singly this cell type specific gene that enriched like to the cell, including cellular response to stress and respond to unfolding protein, respond to oxidative stress. That so that means they really have their special feature, especially related to the HSPC function. Now we as we do the same before, we only using if we do not use HSP gene, only using the other gene, that means the whole genome-wide gene except the HSPG, then we found that this cluster disappeared. Now, where they located? Now they mainly located on naive. So we can see the percentage. So if we do not using HSPC related gene, using all the other gene, we found that the majority of this HSP will mapping on the naive T. That means so majority should be derived from naive after stimulation. Then if we only using HSP high T, then we find that most of the cells also make a cluster. That means indeed the HSP really contributed to this cluster, and we do the confirmation if we use CSF for to do the enrichment. We found indeed that this potentially HSPG high, they express the HSPG, and we are interested in how what factor affect the T cell activation. Then we get one two condition. One is direct stimulated CD4 T cell. Another is that we stimulate CD3 T cell, that including both CD4 and CD8. Now we found that if we direct stimulate CD4 T cell, that's they have the HSPC high T and interferon high T cell. Yeah, and then if we stimulate the CD3, now we found they have very few HSP high T or interferon high T, so they are more likely to differentiate to effective T. Then here we can see the similarly like this is a resting cell after stimulation. If you co-stimulate with CD8, that means then you will have very few HSPC high and or very few interferon high. Then this is consistent. We see the cell subtype like HSP high T mainly derived from the stimulation of the CD4, and also this cluster IN high T is also mainly derived from stimulation CD4. And then there's also signal change for once you co-stimulate CD8, there's a lot of gene expression changed. So that means the co-stimulation changed the not only the cell subset, also the gene expression level or all the T cell subset. One of the intriguing is that we found that if we using co-stimulate just stimulate CD4 co-stimulate with the CD8, then we found that the distribution usually more narrow, but for direct stimulation for more broader. So that means the co-stimulation makes the cell states more similar, reduce the heterogeneity. Then we analyze the cell interaction. We found that in the CD3 and in the CD3 cell, the CD4 and CD8 have a strong interaction. Especially those signals like initiated by CD8 T cell, those they can directly change interact with CD4 cell, and like the cytokine signal and FASALG signal. So that's is FASALG is important for regular T cell. So that means the CD8 can regulate CD4 T cell activation. If without the T cell, maybe explain why without the CD8 T cell, the CD4 respond to stimulate more heterogeneity, but the CD8 they like using the FASLG signal make the cell more homogeneous. Also, if we analyze the stimulation in different factor like cytokine, we found that among all those factors that the resin cell type play most important for the final definition of their activation. Okay, now we talk some more related with tumor immunity. Now we talk the leukemia. For here we have a patient. It's have 4 time point data like at the diagnosis, at the refractory remission, and the relapse. So here we have a reference. Then we can see the cell by a single cell level. We can see diagnosis leukemia cell, then during refractory, finally, it's almost the remission. It's very few cell, maybe very few cell are tumor like. Then relapse now a lot of leukemia cell come back, but they really different from original leukemia cell. Like here we can see that this is original leukemia cell, then this is a leukemia cell after relapse. So that's show quite different. They are quite different, and then their feature are also different. We see the relapsed leukemia cell, they are very strong cell activation and then very higher entropy. That means they are much more active than the original leukemia cell. Then we check the whether those leukemia and relapse cell have with the mutation. Indeed, we found that the leukemia cell with this already have mutation like SM, D, D5A. That means maybe those are leukemia cell specific mutation, but more dramatically, that is the relapse leukemia cell. They also have a specific mutation, like they have specific mutation on IKD1 and RB1. Those are strongly related with cell activity and the usually associated with the leukemogenesis. And then we see that if we cluster those leukemia cells, here is the original the leukemia cell as a diagnosis, then this is relapsed leukemia cell. Then because of before we already developed CCI can project the leukemia cell and develop lineage. Now we found the relapsed leukemia cell project on the earlier stage of the lineage. Now the original diagnosed leukemia, they are more near later stage of the B lineage. That means so the relapsed cell, they have increased stems and they are at a much earlier stage of the this lineage. And then we want to know whether when the relapsed leukemia cell show up. Surprisingly, we found one haplotype. We found it's the relapse specific, and then we check it's whether we can find it in the earlier stage. Then we found that this haplotype could even be identified at a much earlier stage. That means when at the diagnosis. So that means this relapsed leukemia cell, they can be found in the diagonal, but they are extremely low percentage. So the model is that the relapse leukemia cell, they are very earlier times, but they do kind of small fraction of total cell data during relapse. They expanded and become a dominant cell type. And we also found that there are monolithic expression increase during the leukemia progenesis. That usually like once the leukemia development, then there are more monolithic expression. And also we detect the random monolithic expression in that gene. That means those monolithic expression randomly show up in one of the cell, but they are monolithic expression in different region another cell. So that's very interesting. So here we talk another very surprise story, very short story. That means when our collaborator Yi Zhang, he told me he found that they have some sample that showed that potentially the HBV positive patients, they respond to that. I mean, this is a cancer patient. They when the cancer patient they have their HBV positive, potentially they respond to PD-1 more efficiently. Here we show is significant change, but others maybe not change. But here we can show that the PD-1 immunotherapy achieve a favorable outcome in HBV positive non-liver cancer. So then we conduct the analysis on the cell from the HBV patients, heart patients, and the non-HBV patients. Then we found for we can identify the exhausting high T cell, and they are quite different from the normal T cell. And then we found that the HBV plus concentration, non-liver concentration, they have more percentage of exhausted high T. And this may indicate that those cell they have more exhausted T, maybe then usually we know that more exhausted T will make the patient respond to PD-1 immune therapy better. Okay, now we also found that usually the HBV, although they have higher percentage of exhaustive high, but they show the higher cytotoxic score. That means they maybe but they also have a lower cytotoxic score. So that means that this may be because both cancer and HBV make their immune system are more exhausted. The more exhausted, they have more exhausted T, then but PD-1 attack the exhausted T, then make the response better. Then we indeed analyze some cancer patients. We call the ESCC patient cancer patients. They have the they have the different cell type. Here we found that for the HB, they response to the they response to the immunotherapy, and they show like here different we have the HBV minus, HBV positive, and their response to the therapy differently. Here we found that indeed the HBV plus HBV positive patients, the response to the immunotherapy much better. After the therapy, they have increased cytotoxic score, and they have the specific cell subset. We have the CSCR one high subsets, and indeed the some subsets that associated with the better therapy. And now we talk one of the most exciting stories. This is collaboration with Jun Chen in Zhongshan University, Zhongshan University, and in this study we do a lot of single cell study. Firstly we analyze the tumor microenvironment. We found that for the patient, the tumor cell, the immune tumor tumor infiltrated cells, especially the myeloid cells, their number increased a lot in the tumor, and compared with the normal tissue. And they show that means the abundance increase, and also the gene spread change also shows the myeloid cell, they have much stronger cell expression change compared with the other cell types. Like so the myeloid cell play important role. Then we calculate their score, especially the immune inhibition signal. We found that indeed the macrophage, neutrophil, they have much higher immune inhibitor score compared with other immune cell in the tumor. And they like for macrophage, the tumor, they have specific expression gene rich pathway. They make the immune more inhibitory. That and also we especially for the tumor infiltrated myeloid cell, they a lot of subsets change, and one of the gene we call SPA alpha, SIAPA. They show the dramatic increase in the tumor tissue, and also but especially this gene we found that this gene is once it's knockout mouse, its tumor volume is much smaller compared to the wild type. So that means this SBA deletion maybe inhibit the tumor development. So also the different using different cell is showed similarly, and even the challenging and also supported the observation, and they also with for the SBA knockout mouse they have watched lung survival, and this is based on incubation model. Now we talk about using the AOM DS induction model. That is, we found that once you after the induction, the SPA alpha mouse they have very good survival, but wild type some death, and then we found that yes, wild type more likely to develop the more tumor, but the SPA alpha knockout mice they were few tumor. It's based on the statistically it's the SPA knockout mice with much fewer tumor load, much fewer tumor number, much tumor volume, and each tumor is smaller, and the tissue is so similarly now. The wild type more likely to be a tumor tissue, but for the SPA, it's still similar to normal tissue. And we further want to check whether this is caused by the tumor microenvironment change. Then we conduct the single cell assay on both wild type and the SPA minus mouse knockout mouse. And the tumor here we found that for the tumor for the SPA knockout mice, it's have fewer myeloid cell, and but the wild type and has fewer myeloid more T cell. And also we found that the SPA knockout mice they have more stronger antigen prioritization, phagocytosis, and inflammatory response. So also at the subset level, the SPA minus mouse have a lot more specific myeloid cell subsets. Then we check those subsets. One is the we start the macrophage lineage. We indeed found that this here is lineage development from the earlier like similar as then later developed to the tumor associated macrophage. But surprisingly, that is that the TM3 are more likely to are much more strongly enriched in the SPA knockout mice. But here TM2 is in wild type mice. That means this development, the TM3 at a later stage, but the TM2 earlier at least earlier stage than TM2, than TM3. Then we check their expression, found and do a geoterm, found the TM3 with stronger they have more function to kill the tumor, and like they have stronger phagocytosis and stronger lysism, and stronger antigen prioritization. But for the TM2, their function not so immune function not so strong, and but it's wild type specific. In this way, we can say the knockout mice they especially enrich the TM3, but TM3 with much stronger antigen prioritization, phagocytosis, inflammatory response, and chemical activity in the TM2, while TM2 is mainly in the wild type. So this may explain the tumor microenvironment differences. And also another is GMDSA. It still shows similarly as the macrophage. Here the GMDSA is in the knockout mice. It's mainly enriched the GMDSA5. Here it's have more immunology function, while the wild type this is the GMD5 is enriched in the wild type, they are at the earlier stage to know how much more immune function. But for GMD6, they have stronger immune function. Then, yeah, also indicates the immune environment different. And also, this is we talk about the microenvironment difference. But how the environment shaped? Then we also analyze the macrophage function. And for the knockout mice, knockout macrophage, SBAF knockout macrophage is have stronger phagocytosis, while that means that maybe explain why the knockout mice they have much fewer tumor when you induction the tumor, and then when the deletion of the macrophage, and we found that it's we're change the tumor size. That means this macrophage play important role in the in the tumor development, and that is the macrophage itself. But we know the whole microenvironment or tumor microenvironment right changed. Then we and the T cell, we found the knockout my specific T cells, they are enriched the functional immune function, and either CD4 or CD8, they knock out the deletion of the T cell, either CD8 or CD4, then we'll change the tumor volume. Then means they play both T cell CD4 or CD8 T cell play important in the tumor development, and then later we indicate found that that's how they should, because we know T cell itself don't know the expressions by alpha, how they change it. So I mean, may potentially influenced by the macrophyloid, macrophage or neutrophil. Then we found indeed the TAM and GMIBC, this myeloid cell, they have interaction with the either CD4 or CD8 T cells, and the signal like CCL, there are a lot of signals that directly send a signal to the from macrophage to the T cell, and then we found that the proliferation for over the are different for especially for the SBAF knockout mice, they can more strongly influence the T cell proliferation, and then either using the cell line or using the TM. And we further to check the crosstalk between the T cell and the macrophage, and indeed the tumor macrophage have signal on CD4 and CD8, and then those we found that the knockout mice, they can for the TM, they can more strongly recruit the T cell. Then that means we can get that in the knockout mice, they have more recruit more T cell, and also they play also higher T cell proliferation. And then we found that the cytokine, they play different, some like here the different cytokine are playing different role in recruiting the T cell, like especially here like CCL3, they have stronger effects, and here it's CCL8, especially for the CCL8, especially for recruit the CD8 T cell. And how CCL8 influence the T cells, and we indeed checked the potential target that we using the inhibitor like found SYK inhibitor can strongly reduce the CCL production expression. Then that means because SYK are high expressed in myeloid cell, and that means they once they have yes, stronger expression can induce the more T cell in the knockout. Okay, and finally, we check the SPA because we know that currently most efficient immunotherapy is PD-L1, PD-1, and here we checked that we found that if we have the SPA knockout mouse, then it's once it's treated with PD-L1, it's the tumor volume decreased, and so that means potentially if SPA knockout should is a better target for immunotherapy, for joint immunotherapy with the PD-L1. Okay. Yeah, this is almost finished, but we also just started to add some spatial transcriptomy, and like here, this is the normal clone tissue, and this is the CRC tumor tissue, and we did find they are quite different, and yeah. So this is ongoing work, but we do not talk much, but at least we know that the different cell type have different species location on the tissue. Okay. Finally, we know that although single cell RNA is very efficient, but still it is only usually capture one modality. We still need more single cell multi-modality technology to detect get more modality to provide more comprehensive information. And also for this, there are a lot study like especially spatial tracheotomy. They can really provide more information, but they can either they are much limited number of reads or lower resolution still not provide enough information, still need to be developed with maybe other technologies by single cell sequencing or integration to provide more information. And another thing is that direct contribution of the single cell sequence to the precision medicine is still limited. Although we already said, oh, this single cell really will help a lot, but maybe we still need to find a way how to really facilitate the precision medicine. And then the single cell RNA-seq or single cell other single cell technology develop very quickly, but in the future, like with the cells with 3D information is still very important, because if you only know the state but you don't know where it is, still could not get the complete information, still not really get the biology insights. And another is that the single cell omics for its potential, because we always think if we sequence all the cell, then we can tracing back, because then we can tracing back to where it started. That means we can use a single cell sequence to reconstruct the whole organism. That's sounds good, but it's still not, especially if the sequence area is high. For normal cell, it's not easy to really construct the whole organism lineage. So finally, we know that each cell is have different states and different space. Like this onion epithelia. It's this plot in 100 years ago, we know that each cell are different, and its cell cycle or some other cell states is quite different. So each cell is unique. then it's occupy extrusion operation in space and with a specific error in its copy the genome, then it will have a spec feature. Okay. Lastly, we'd like to thank our collaborator. For the genomics, we use the single cell policy collaborated with Niting in Fudan, and SEGSIG is collaborated with Xichen, and single cell graph autoencoder is with Zhang Zhen, and also Keji is my advisor when the work from the work in Edge. For the last most SPA for this is important collaborator, that's Jun Chen. He already work on this field a long time, and for the leukemia is collaborate Teng Tao. Okay.